afternoon, everybody, and welcome to Mindset. Today you've got me, Kathy, and
Indiana, as always.
We're going to be working through life sciences. All right, so life sciences, and we're going to have questions. And remember, it's all paper one. So with paper one, we are looking at genetics. We're looking at DNA and DNA replication and protein synthesis. We're looking at meiosis, which is easy. I'm sure you won't be asking about that. And then also the whole diversity and evolution and blah, 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 and apes and primates and
all those
kind of things. Yeah. Okay, so feel free to ask. We've already had hundreds of questions here on just protein synthesis, so I think that's the question we're going to start on. We have a caller, am I right?
I think so, but before we get going, I just want to let everyone know that please join us on our Facebook page, facebook.com/learnextra with an X. Chat to us, and please try and send in relevant questions to what we're talking about. We'll try and get to as many as we can, but obviously we have a short space of time. Also join us on Twitter @learnextra, as well as PepText. Let's get going, and I think we have a caller. Do we have a caller?
Not yet. Okay, so you know what, people, while we're waiting for our caller to come online, what I want to do is explain something very, very simple to you, and just the very basics of what protein synthesis is about. Now, you must remember, every single one of us, now if you think of this logically, then this whole process makes sense. So, every single one of us has a set of proteins. Indiana sitting next to me here and we're both girls. So we will both have an XX chromosome, okay? We've both got 10 fingers and 10 toes and 2 eyes and a nose and a mouth, blah, blah, blah. That makes us human, all right? We both have 23 pairs of chromosomes, and at some stage, our body needs to make new cells. All right, so mitosis will take place to repair, to replace any cells that are damaged, and then to make new cells, so for growth. When all of this happens, the DNA has to replicate, because I've got to make sure that the DNA in this little cell, when it makes a new cell to repair it or replace it, I need to have the same genetic material in there. Every protein in our body, the recipe is written inside the nucleus on the DNA. So the DNA, if you think about an analogy for it, it is just one huge big recipe book for you. And you know, if two people make chocolate cake, unless they're using the box version, but if they make chocolate cake from scratch, even if they're using the same recipe, the chocolate cake that, if India and I both did one, would be completely different to, she may use cocoa icing and I'll use chocolate icing, and maybe I leave mine in the oven a bit too long.
I was going to say, that would be me. I would be the one that would forget to put, the flour and the eggs in and comes in concave.
Yeah, but exactly. So it would be different, even though the basics were the same. So our proteins are not the same. If I wanted to give her a kidney, I couldn't. Why? Because her body would reject, would say, this is not part of our clan here because the proteins are different. Which says we need a huge recipe book for the proteins. So when we have our DNA, we're going to, and I'm going to do just a very short table here, and our caller's on the line. What's your name, doll? Hello?
Hello.
What's your name?
My name is Christina.
Christina, where are you calling from?
I'm calling from Carltonville.
Oh, from Carltonville's not too far away from Joburg. So you're also sitting with this heat wave going at the moment, hey?
It's very hot in here.
And here in the studio, we're sitting with a temperature of 27. So, yeah, by the time we finish the hour and a half, we're going to be sitting here sweating.
My face is going to melt off.
We'll both have lost like 5 kilos in water. But anyway. Okay, Christina, apparently you're battling with protein synthesis, hun?
With protein synthesis, as a term of the stages from 1 until the last stage of this process called translation.
Okay. What you've got to remember. The line, I'm getting echoes here, but what I want to try and explain to everyone here about protein synthesis is we need to make the proteins. The recipe is written here on the DNA. Okay? So that's where the recipes are going to be. And those recipes are written in little blocks of 3. 1, 2, 3. So this is what we've got here.
Maybe you should just turn your TV.
Yeah, Christina, try and turn your TV volume down just a little bit, hun.
Okay.
So there's an echo.
Then the two of us can actually hear you. You see, okay.
See, I don't like the sound of my own voice, Kathy. I hear it, I'm like, oh, it's terrible.
All right. So there, now remember, on the DNA, you've got a double strand. Okay. And sitting on that double strand, if we break that strand open, we're going to have this here, which is going to be a code on. It's got just 3 little pieces that's now going to, well, it's not the code on, it's going to form the code on. Okay, here, the second strand that we have is going to be the messenger RNA. Now remember, the difference in RNA and DNA is DNA is a double strand, RNA is a single strand. Okay, DNA is the code of life. That is you. Everything that's in that DNA, on those 23 pairs of chromosomes, that DNA is you. Very, very unique, special individual. The messenger RNA's job is to take the DNA, make a sort of a copy of it, and then take that to the ribosome to make protein. So it's getting the recipe piece here in little bits of 3. So that's your messenger RNA. And then at the ribosome, which is where protein, I mean, you learnt in grade 10 that the ribosome was responsible for protein synthesis. So at the ribosome, we're now going to have our tRNA, which is our transcription RNA. Okay, so our transfer RNA. So now what happens? The messenger RNA comes along. Okay, wait, let's just first do this here. Let's say, now remember, we've got thymine, we've got adenine, we've got guanine, and we have cytosine on DNA. So let's go T, A, G, C, C, A. Okay? And that is what we are going to code on. So the messenger RNA says, oh, okay, fine, let me come along here, and it codes on. This is my codon, and it will code on to TAG, but it's going to be the complementary base. So what is the complementary base to thymine is adenine, because it's always adenine to thymine and guanine to cytosine. They always pair up. You will never ever get thymine and cytosine together because they are both they're both pyrimidines. You will never get guanine and adenine together because they are purines. The purines and the pyrimidines, they don't go together. They've got to be one purine, one pyrimidine, and that's what bonds. Okay, it is always adenine to thymine, always, and guanine to cytosine. Nothing else. Okay, so what you do whenever you get a question on on DNA and protein synthesis, you write A equals T and G equals C at the top of your page, always. Okay, so thymine, the opposite, the complementary is going to be adenine. The complementary to adenine is going to be thymine, but in RNA, there is no thymine. It is always uracil. Okay, and then guanine would be cytosine. So this one here, this little one here is now going to, that's its complementary base. So the difference between DNA and messenger RNA is that they are always opposite. Okay, because it's the complementary bases. Now, here, your transfer RNA has to now take this information that the messenger RNA has given, and it's now got to do what is on the DNA. So it says, okay, not a problem. This is what we are going to do. And your transfer RNA now does this. And it says, okay, we're going to have no thymine, but uracil, adenine, and guanine, which tells you what? That your, let me just make this move up a bit.
Clever thing, this Wacom.
Yeah, Wacom.
Wacom.
Okay,
Just whack
between this one and this one, okay, what do I have? They are opposite as well.
Yes.
Okay, but look at this. They are the same as the DNA, except that because it is RNA, you are always going to have uracil instead of thymine. Okay, and with DNA, you are going to have thymine and not uracil. You understand?
Okay.
Okay.
Yes, I understand.
Okay, so now look, the DNA and the messenger RNA are opposite. The transfer RNA and the messenger RNA are opposite. But
Perfect.
your DNA and your transfer RNA are the same. Does that make sense?
Yes, it makes really sense.
Now that's all you have to do in an exam is remember this. If you remember this, this is your recipe for success. Okay, where protein synthesis is exactly. So now, Christine, let's have a look here. Here we've got a question that says, the diagram below shows the process of protein synthesis. Let me just get blue here. Okay. First thing you do in an exam, and I mean if you've ever watched me, and for all of those that have watched me before, you will know. I've said if you get a diagram, step one is you write the labels on here. Why? Because it's the same as your brain pulling out the file that says, protein synthesis, and it takes the file out. So now you're ready to learn about it, and you're ready to get full marks. So here we go. If we look here, Christine, what is this M? What is this thing here? This double twisted thing. It's a
It's a DNA strand.
That's right. It's a double DNA strand. Okay.
Yes.
And if we look here, this piece here is the one side of the DNA. You see that? Because it's now broken apart. So this is one strand. Okay. Then N, if we look at N is actually this part here. What's happened is it's come and it's turned over. So that's this here. So this one and this one are the same. So N is going to be our messenger RNA.
NA.
Okay. And remember, the messenger RNA is going to be opposite to the DNA. So you see what I'm doing? I'm writing on my diagram so that I can see, this is what I have to answer. Z is going to be a codon. How do we know that? Because it's referring to 3 little bits. So we call it a codon or a triplet base.
Okay.
Okay, both are correct. I like codon because you code on. It codes onto the DNA and it says, okay, I've got you now. And then if we look here, Q, this part here is going to be an amino acid.
Acid.
Okay, that's my amino acid. Because remember, lots of amino acids together, if I have lots and lots and lots, it's going to give me a protein. All right,
Okay.
R is a ribosome. And a ribosome is an organelle in a cell that is responsible for protein synthesis to make proteins. All right?
Okay.
Okay. What are we going to have here? And that's still my codon. And Q would be the amino acid, and this here would be my anticodon. Okay, so this X is the same as Z, so I don't know why they would have labeled it like that. But then we also don't know why they label things the way they do in an exam. Sometimes they do make a little bit of an error. Okay,
Yes.
and then this part here, oh, hang on, this is your amino acid. Kathy's wrong. Amino acid, although these two are still the same. And this here would be the ribosomal RNA, or where's my transfer RNA? Let me just see. This would be my transfer RNA. All right, so having had all of this now, Christine, now we start and we say, identify compound M and R, organelle R. So compound M is the DNA, all right? And organelle R is the ribosome.
Ribosome.
Cool.
Yes.
Okay, we are just going to read the question, then I want you to think about the question because we are going to be going for an ad break in about 20 seconds. So it says, write down the sequence, and that's the operative word, of the first 3 nitrogenous bases on the DNA that led to the formation of Z. So think about it, and while we go for an ad break, and when we come back, we'll go through it.